So, question 5a, the nucleus and chloroplast of a plant cell both contain DNA. Give three ways in which the DNA in a chloroplast is different from the DNA in a nucleus. This is quite interesting because this ties into a theory called endosymbiosis. You may be interested to look up on that theory about the origin of chloroplasts and mitochondria within eukaryotic cells. Anywho, skipping on to that, let's get down to it. So think of this question when they start talking about DNA in chloroplasts. It's basically the same as the DNA you find in prokaryotes, which gives us a link to what a chloroplast was in a past evolutionary life many, many years ago. So how is it different from the DNA found in a nucleus? So what do we want to say? We've already said them. It has no histones. Of course, the DNA in a nucleus has histones. So has no histones. We're going to write the same five things down, right? Or four things. Has no histones, has no introns. What else can we say? It's got no junk DNA. It has circular DNA. So the chloroplast has its own circular DNA, even though the rest of the plant cell has linear DNA. The nucleus of the plant cell contains linear DNA. The chloroplast has circular DNA. That gives us a hint to its past, evolutionary past. So there we go. I've mentioned three things. Lovely jubbly. And then we could probably say the DNA inside the chloroplast is going to be shorter because it contains fewer genes than the DNA in the nucleus, which contains the genes for the whole of the plant, whereas the DNA in the chloroplast is just the genes for the chloroplast. But there you go. Any two of those three will do. Oh, you had to say three differences. Well, it's lucky I gave three differences, right? So there we go. I gave three differences anyway. That's me thinking it was a two marker. B, some DNA nucleotides have the organic base thymine, but RNA nucleotides do not have thymine. RNA nucleotides have uracil instead of thymine. You know, give one other difference between the structure of a DNA nucleotide and the structure of an RNA nucleotide. So what's the other difference? Think about your nucleotide. Both contain a phosphate, both have a pentose sugar, both contain bases. However, we've just been informed the RNA base has U instead of T. What's another difference? Well, it's right here. It's not in the phosphate, it's with this pentose sugar. What's the name of the pentose sugar in RNA? So we would be like RNA has ribose sugar, whereas DNA nucleotide has deoxyribose sugar. And that's really the only other difference with the nucleotides. There's only two differences. One is the uracil base, and the other is on the sugar, there's a difference. However, there is difference between RNA and DNA, as in DNA is double-stranded, RNA single-stranded, etc. But this was specifically about the nucleotides, not about the strands of RNA and DNA. Hokey cokey, let's move on. The diagram shows the tRNA molecule. So this is a transfer RNA molecule. And we're going to be asked name structures W and X in the diagram. Well, I'm pretty sure you'll probably remember the structure X. This is what binds by complementary base pairing to the codon on mRNA at the ribosome. CUC actually codes for leucine, the amino acid, and it is known as the anticodon. So this is the anticodon. It's where the tRNA molecule temporarily hydrogen bonds or binds by complementary base pairing to the ribosome. Now, name the structures labeled W and X in the diagram. So W, what is W? What attaches to W? Well, W is where it's going to carry the amino acid. Obviously, this will, the tRNA will bugger off and leave leucine behind, and leucine will be bonded by a polypeptide, by a peptide bond to another amino acid. So this is being brought down. What we call W is the attachment site or the binding site. So it's the amino acid binding site. It's where the amino acid attaches or binds or bonds to the tRNA. Of course, the amino acid is left behind when tRNA leaves as the next tRNA molecule comes. Okay, and that is going to get you a mark. You needed both to get a mark. That's quite difficult, actually. I guess anticodon everyone would have got, but the amino acid binding site may have Slipped a few people up. Right, let's go down. Let's finish this off. So part D, not all mutations in the nucleotide sequence of a gene cause a change in the structure of a polypeptide. Give two reasons why. So why a mutation is a random change in one of the bases. So G could become C, C could become T, but then you get deletions where, say, T will disappear or G will disappear, and then you get insertions where extra bases are put in. But what kind of mutation will mean that nothing changes? The polypeptide retains its 3D shape and is still functional. So what would that be? Of course, we could get a silent mutation. What is a silent mutation? This is, remember that, I'm just going to put the explanation. DNA is degenerate. DNA is degenerate. So each amino acid has more than one code, right? So each amino acid has more than one codon. So changing the triplet code may not change the amino acid. Let me give you an example. So for example, just so you can understand it, CCC codes for glycine, right? Imagine there's a mutation. One of the C's is turned to a G. CCG also codes for glycine. So essentially, we're going to get a mark for saying, well, just because there's a mutation, it might be a silent one. There's more than one triplet for each amino acid, and that will essentially get you those 2 marks. Lovely. Now we need another reason. Where could the mutation happen where it wouldn't affect the amino acids? Of course, the mutation could occur in non-coding DNA in introns. So if it occurs in the non-coding DNA, that's not going to do anything. So if the mutation occurs in non-coding DNA, they do not code for amino acids. So it's not going to affect it. So we have to say it could occur in introns, and these don't code for amino acids. Boom, we get all the marks. 2.1, what is a monoclonal antibody? So that's the first part of the question. What's a monoclonal antibody? Well, these are identical antibodies. How do we say that in A-level language? Well, what is identical about the antibody? From one antibody to another, what is it that is identical? It's the tertiary structure, so the 3-dimensional shape of the antibody that's identical. So they will all bind with the exact same antigen, a bit like a lock and key theory. The antibodies are all complementary to the same antigen. So how do we word that? We say monoclonal antibodies are antibodies with identical 3D or tertiary structures. Now, another definition of monoclonal antibodies is that they are all produced by clones or identical plasma B cells. So all these antibodies are made by plasma B cells or clones, identical cells producing the exact same antibody. Don't say something like monoclonal antibodies have the same DNA. They don't have DNA or genes. They're actually just a string of polypeptide. They're just some polypeptide chains, aren't they? They're just a sequence of amino acids, but they have the identical sequence, so the identical tertiary structure. Now, the second question, 2.2, after a disease is diagnosed, monoclonal antibodies are used in some medical treatments. Give one example of using monoclonal antibodies in a medical treatment. So if you had lung cancer, some of the cells in the lungs would be cancer cells. Okay. Now, they would have antigens that may be slightly different to the other cancer cells. If you wanted to target them with a drug, with a chemical that kills those cancer cells, you can attach that drug to a monoclonal antibody that is complementary only to the antigen of the cancer cell. So it's very specific in the cells it targets. So monoclonal antibodies can be used to carry medicines or drugs to specific cells. You might have a disease of the liver and you want this medicine to only go to the liver and attach to liver cells and affect those. Well, you would then use a monoclonal antibody that is complementary to the antigens on liver cells. Here I'm drawing a little cancer cell. So let's draw a normal healthy cell. This will be a healthy cell. So I'm going to put an H in the middle for healthy cell in our lungs. And then next to it, I'm going to draw a cancer cell. Now, because of the mutations, etc., it's got slightly different antigens to the healthy cell. This is great because this means I can take a drug that will kill and destroy cells, and I will only deliver it to those cancer cells and not the healthy cell next door. So I would attach a drug, which I'm going to use a green to attach a drug to these monoclonal antibodies, these pink ones here. They will only attach to the cancer cell because the antibody is only complementary to the antigen on the cancer cell. The tertiary structure is only complementary, so will only bind to the cancer cell. Those little green squiggles, those are my artistic impression of a cell-killing drug. Well, let's get down to the main question here. 2.3. Describe the role of antibodies in producing a positive result in an ELISA test or ELISA test. How do you say that? Anywho, the E in the ELISA test stands for enzyme. The L stands for linked. Sorry, I just realised my microphone was sounding a bit tinny there. The audio was a bit messed up. Hopefully I've fixed that now. So the E stands for enzyme and the L for link. So there's a little clue there. The ISA stands for immunosorbent assay, but you don't need to worry about that. Just try and remember the E is enzyme and the L is for link. Then you'll have a bit of a clue, a trigger to get you started. Now there's two types of ELISA test, and these are really good at searching to find out if you have a certain type of disease. We have the direct ELISA test and the indirect ELISA test. I'm going to go through the direct ELISA test first of all. Okay, so with the direct ELISA test, you can take some sort of beaker or some sort of well, and you're going to immobilize some antibodies, monoclonal antibodies, at the bottom of the well. These monoclonal antibodies are complementary to the antigen we're looking for. So imagine we're searching for some sort of flu virus and we know what the antigen is. We can make monoclonal antibodies that will bind to the antigen. We immobilize those monoclonal antibodies at the bottom of a well. Then we take the patient's blood. We only really want the blood plasma because it's clear, and we can use a centrifuge to get that. You don't need to know that. But we add the patient's blood plasma. Now let's pretend these little purple diamonds, these are the antigens to the virus we're searching for, or the bacteria we're searching for. These antigens will bind to the immobilized monoclonal antibodies. Now, in between each stage here, or some of the stages here, we really need to wash. And what do I mean by washing? Well, washing is going to remove any antigens or antibodies in the blood plasma that are not complementary to the immobilized monoclonal antibody. So it's important that you wash so you don't get any false positives or any inaccurate test results. So if the antigens are not complementary, they will be removed, and later on that's going to help us find out the person doesn't have the illness or doesn't have the antigen, the target antigen we're searching for in their blood plasma. So what's the next step of this test? Let's pretend they do have the antigen and they're all bound to the immobilized antibodies. Now what we're going to do, we're going to get another enzyme. This is the second enzyme. And in both the direct and indirect tests, we're looking at the direct first of all, but in both tests, the second enzyme is the one that has the second antibody is the one that has the enzyme linked. So it's always the second antibody you add that has an enzyme attached to it. So the second antibody with the enzyme attached is added. Now this antibody that I'm drawing in green here is a Y shape with the little enzyme like a Pac-Man on the end. This antibody is also complementary to that target antigen, and it will bind to it. Now we wash it again because this is the most important washing. We need to wash this beaker or this well again because if that purple diamond, that antigen, is not present, the second set of enzymes will wash away and we'll only be left with the immobilized antibodies, right? That's very important that we wash it again because we don't want a false positive. So what we're going to do here now, let's pretend again that the antigen was present. So the second enzyme with the enzyme linked has attached as so, like I'm drawing here with the little enzymes at the end. The second antibody, also complementary to the antigen, has attached. What we're going to do now, what do enzymes react with? We're going to add a substrate. Now that substrate is going to have a complementary shape to the tertiary structure of the active site of the enzyme. They're going to fit like a lock and key. They could bring that in if it's a paper 3 question. And the substrate will catalyze that reaction, weaken the bonds and break the substrate. And this substrate will change color and give us a color change. And then we will know, aha, the person had the target antigen. They are infected with that disease. So we're looking for this target antigen. Potentially it's from a disease that we're testing to see if the patient has the disease. And if we get a color change when the substrate binds to the enzyme that's attached to the second antibody, boom, we know they have the disease. So I'm going to summarize this direct ELISA test into just 4 key bullet points. So summarizing the stages of the direct ELISA test. Number 1, the target antigen is going to be added from the patient's blood, if present, to the immobilized antibodies. Because they're complementary, they're going to bind together. Step 2, the second antibody is added with the enzyme link. This will also bind to the target antigen. Then we're going to wash it. In case they don't have the antigen, everything else will be removed except the immobilized antibody, and we won't get a color change. But if they do have the antigen, in the 4th step, when we add the substrate, it will bind in the active site of the enzyme, it will be catalyzed the substrate, and it will give us a color change, proving that the target antigen was present in the patient's blood plasma. And that is the summarized direct ELISA test. We're now going to look at the indirect ELISA test and what that looks for and what the differences are. Now, first, a quick reminder why it's important for step 3 to do the washing here. If you wash the sample, if the person doesn't have the antigen, i.e. they don't have the disease you're checking for, then the enzyme will be removed when you wash, and then you won't get a color change, and you'll know they don't have the disease. If you forget to wash in between step 3 and 4, then the enzyme will be there regardless of whether they have the antigen for the disease, and when you add the substrate, you'll get a color change, and you'll get a false positive where you go, you have the disease, but they don't really. All right, let's move on. Okay, the key difference with the indirect ELISA test. So the key difference with the indirect ELISA test is that we are searching for the presence of an antibody produced by the patient's immune system in response to the target antigen. We're not actually searching for the antigen. So this might be a virus where it's really hard to find the virus. It's so small, or something like that. And actually, it's much easier to look for the antibodies because a patient who has the disease will produce large amounts of the antibodies. So it might be a lot easier. And this is what they do with HIV when they're testing you for HIV. They actually look for the presence of an antibody in the patient's blood. So they start, instead of immobilized monoclonal antibodies, they start with immobilized antigens. We then add the first antibody that is complementary to the HIV antigen. Now, if you have the HIV antibody in your blood, it probably means you've got HIV, and that HIV antigen will bind, will bind to the immobilized antigen. Now we wash it to remove any antibodies that haven't bound, and if the person is HIV free, then they won't have any antibodies bind to the antigen. So we do a washing here. The next step is to add the second antibody, and just like in direct ELISA, what you have to remember, because it will be on all the mark schemes, the second antibody is the one with the enzyme attached, the enzyme linked antibody. So we add that, and now, unlike in direct ELISA, it's not complementary to the antigen, it's complementary to the first antibody. So you can see here the green enzyme linked second antibody I've just drawn binds to the first antibody that's bound to the immobilized antigen, and then we add a substrate. If there's a color change, the person has it. Of course, we wash again before this. We wash it to remove unlinked second enzyme antibody. But after the washing, if there is any enzyme linked antibody left, then the substrate will change color, proving that that antibody is present for HIV. Welcome to Let's Get to the Marks, going through a Benedict's test and reducing sugars A-level biology question. Don't forget to pause the video, have a go, write your answer on scrap paper, then check the answers. Figure 2 shows the structure of 2 alpha glucose molecules. On figure 2, draw a box around one chemical group in each glucose molecule used to form the glycosidic bond. So, where does the glycosidic bond form between these 2 glucose molecules? Well, a condensation reaction takes place. You've got to ask yourself, what gets lost or removed in a condensation reaction? Of course, it's H2O that's removed. But what 2 groups does it come from? It comes from these hydroxyl groups. The OH groups are called hydroxyl groups. So you could circle both of them, or you could just circle an H on one and an OH on the other. So either of these are correct and acceptable. The mark scheme's first point is to circle both chemical groups, which are the 2 hydroxyl groups. But of course, you only lose one OH and 1H. So you could just circle an OH and then an H. 3.2. A precipitate is produced in a positive result for reducing sugar in a Benedict's test. A precipitate is a solid matter suspended in solution. A student carried out the Benedict's test. Suggest a method other than using a colorimeter that this student could use to measure the quantity of reducing sugar in the solution. So, once again, going back to this, a precipitate is a solid that forms in a liquid. So, with the Benedict's test, you're using copper sulfate and sodium hydroxide, which has a blue color, and you get a red precipitate. So how could we, without using a colorimeter, how could we measure how much sugar was in there or get some sort of idea, a comparative idea? Well, we could filter off the solution, dry the precipitate carefully with paper towels, and then weigh it or measure its mass on a balance. And therefore, if you had two different solutions, the one with the higher mass of red precipitate is going to be the one that has the most sugar in it. So we could use that as a quantitative measure of the glucose concentration, or sorry, the concentration of reducing sugars in the solution. So what do you get the marks for here? One is for filtering the solution off and then measuring the mass. Right, let's move on. In an investigation, a student wanted to identify the solutions in two beakers, A and B. She knew one beaker contained maltose solution and the other beaker contained glucose solution. So let's label one of these M for maltose and one G for glucose. Both solutions had the same concentration. She did two separate biochemical tests on each beaker. Test 1, she used Benedict's test on each beaker to see if there's a reducing sugar. Test 2, she added the enzyme maltase and then repeated the Benedict's test at a temperature of 30 for 5 minutes. Now we can see that beaker A is the one with just the reducing sugar, and beaker B, or the glucose, and beaker B contains the maltose. Now what's happened with beaker B? It was red, but then it goes dark red. Why? Why in test 2 does beaker B go dark red? Well, we added maltase, and that's an enzyme. So let's find out. Explain the results for beakers A and B in the table. Well, the reason why beaker A goes red with both tests is because it shows the presence of glucose, because we've done the Benedict's test and glucose is a non-reducing sugar. However, with beaker B, it goes dark red on test 2 because the maltase, which is an enzyme, hydrolyzes the glycosidic bond in the maltose, snapping it into two glucose molecules. So if you have the same concentration of maltose and the same concentration of glucose, if you hydrolyze the maltose, you'll double the concentration of the glucose because each maltose molecule is made of two glucose molecules. So although you might think you have the same concentration of maltose as glucose, each maltose molecule is made of two glucose molecules, so you have double the concentration of glucose in a maltose molecule. It's made of 2G. Right. Let's move on to the next part of this question, as we have solved that one. So you get 2 marks here, which is nice. Don't forget to hit the like and subscribe, people. It really helps the algorithm, the YouTube algorithm, get my stuff out to other people. 3.4, use a colorimeter in this investigation, or use of, would improve the repeatability of the student's results. Why? So what does a colorimeter measure? It measures absorbance, or you can set it to measure transmission. And that means instead of just looking and going, has that gone red? Is that dark red or is that medium red? You actually get a quantitative result. You get a number so you can judge and compare fairly two different concentrations. Whereas otherwise you're using a subjective decision when saying, is this red darker than that red? That's down to people's eyesight and down to people's opinion. We call that a subjective result or evaluation. We don't really want that. So you get a quantitative result with a colorimeter. So in test 1, the student used a measuring cylinder to measure 15 centimeters, that's the measurement, from a beaker. The measuring cylinder gives a volume that has an uncertainty of plus or minus 1 centimeter cube. She used a graduated syringe to measure 5 centimeters. So that's a second measurement using a different piece of equipment. So we have a measuring cylinder and graduated syringe. The graduated syringe gives a volume with an uncertainty of 0.5 centimeter, plus or minus. She mixed these volumes of liquid to do a biochemical test, calculate the percentage area for the measurements used to obtain the full 20 centimeter cube mixture of the solution from the beaker and the Benedict solution show you're working. We need to treat them as two separate, then add them together. So for our measuring cylinder, we had an uncertainty of 1. We're going to divide that by the measurement. So the percentage error is 1 over 15 times 100. So we can do that now, 1 over 15 times 100, to work out the percentage error of the measuring cylinder, or we can combine it with the percentage error of the graduated syringe. Now, the graduated syringe has an uncertainty of 0.5, and the measurement taken was 5 centimeters. So we're going to do, don't need the C there, we do 0.5 over 5. We can add these together if we like, and then we can times 100, and that's going to give us our answer. Dead easy. So if you do that, you get an answer of like 16.66 recurring. You could round that up to 16.67, but there's a whole range up to 17% they give you. And there we go, just a quick little video. Sharing's caring, hit the like, punch up the bell, subscribe. Welcome to Let's Get to the Marks, going through an A-level biology question on the index of diversity. Let's get into it. A scientist identified and counted the invertebrate species present in samples taken at 2 sites in a river. The scientist also measured the rate of water flow at each site. His results are shown in table 2 and 3. We've got the name of the invertebrate species here, and then we've got site 1 and 2. And this shows the number of each species at site 1 and 2 on the river, different places on the river. Total number caught 48 and 45. Then we've got the index of diversity, which is 4.7 on site 2, which is quite high. And the rate of flow of water seems to be 30 to 60 at site 2. So the flow of the water is faster than 1 to 14 at site 1, and we haven't got the index of diversity at site 1, so we can all guess what we're going to be asked here. Complete table 3 by calculating the index of diversity at site 1. We've got to use n over n minus 1 over the sum of lowercase n minus 1, and that will give us the diversity. Okay, so what does the big N mean? Well, the big fat capital N, what does that mean? That is the total number of organisms. So big N is equal to 48 at site 1 and 45 at site 2. I suggest you pause the video, grab some scrap paper, grab your scientific calculator, have a go at this question, and then unpause and watch me calculate it. Okay, so total number of organisms is big N. Little n is the number of organisms in an individual species. So for example, 17, 6, 0, 12, 13, those are all the individual species. So what we need to do is big N multiplied by big N minus 1 over the sum of all the little n's multiplied by little n minus 1. Okay, so D equals 48 multiplied by 48 minus 1. So essentially we're doing 48 times 47, and that equals 2,256. Now we need to do n times n minus 1. So we've got to do site 1, 17 times 16, which is n minus 1. And then we've got to add that to 6 times 5, because we're doing 6 times 6 minus 1. And then 12 times 11 and 13 times 12. We don't need to use the 0. And if we bash that in the calculator, we get 2256 over 590. We get 3.823, something along those lines. You're going to round down. I like to go to 3 significant figures. The mark scheme accept 3.8 or anywhere between like 3.8 and 3.4 or something like that. So I'm giving 3.82 is what came out in my working out. So there you go. That's going to get you the mark. A lot of effort actually for 1 mark, those index questions. Right, 4.2. Explain why it's more useful to calculate an index of diversity than record species richness. So hopefully you know what species richness is. Species richness is the number of different individual species living in an area or habitat. An index of diversity doesn't just take into account the number of species, but it also takes into account the relative abundance of each species, something known as species evenness, the relative abundance. So you might have field A and field B. Field A might have 5 species, but they have 10 of one species and then 1 of 4 other species. And field B might only have 3 species, so 2 less species, but they have 30 of each species. So which one is more biodiverse? We can't really say field A is. It's only got 1 member of each species. So what we're doing there is we're taking into account not just the number of species, but the relative abundance as well. And this takes into account the fact that you may have a couple of really large populations or some really small populations. And that all contributes to the overall index of diversity. The higher the index of diversity gives us an idea of the area that is more biodiverse and more likely to survive drastic changes to that environment. Right. 4.3 suggests how the scientists measured the rate of flow in the river. Well, a good idea would be, here's your river with the arrow showing the flow of the river. What you should do, what does rate mean? Rate essentially is speed. And how do you calculate speed? Speed is distance over time. So you'd measure out like 10 meters of the river, put a floating marker at the start, get your timer out and time how long it takes for your marker to reach the end of that set distance. Might be 10 meters, might be 50 meters, whatever you want. So you get your stopwatch out and calculate how many seconds, and then we can do speed equals distance over time, or rate in this case is distance over time. So measure the distance, divide it by the time. There's your mark. And there we go. There's the answer, guys. So check how you did. Obviously, it's best to pause the video before I start waffling out the answer so you get a chance to write it down on scrap paper. 4.4, use the information in table 2 and 3 to suggest and explain a reason for the difference in the number of slate drake mayfly at the 2 different sites. So site 1, we've got 0. Look here, we've got 0, so no slate drake mayfly found. And at site 2 there were 6. What could be the reasons? Well, look at the index of diversity. It's 4.7. We know site 1 was 3.8, so there's a higher index of diversity. How does that help the mayfly, there being a higher index of diversity? There was also a faster flow of the river. So first of all, site 1 has a lower biodiversity, or we could say the opposite and say site 2 has a higher biodiversity. So if it has a lower biodiversity, there's probably less prey. I wouldn't bang on about, I've written less habitats, I wouldn't bang on about that really. It doesn't tell you that. It tells you that there's less prey, there's less organisms there, maybe less things to eat. Site 1 has less water flow, so the water is flowing more slowly. This means it's less oxygenated, so there's less oxygen in the water, which means there's less respiration. It means the organisms are less able to produce ATP, and that will get you your second mark. Lovely jubbly. So less prey, less available food sources, as there's a lower index of diversity, and then less water flow, so less oxygen. It's important when there's sampling that it's standardized. So this is just asking you, what are some control variables? So you want to take your samples from the same area and at the same time each day, because certain species may come out more at different times of the day. So you've got to do it at the same time each day. Use the same size net or the same size beaker, or however you're capturing these things. Collect the same volume of water. Yeah, maybe not. You don't need to collect the same volume of water, but same position along the river, same depth of sample. Well, you're probably doing it off the surface to catch mayfly and stuff, so you don't really need to do it at the same depth. But if you were trying to get fish, then maybe you'd do it at the same depth. And that's the end of this question. So don't forget, sharing's caring. Hit the like, subscribe, punch up the bell, whatever. Let's get to the marks.
You're still here? It's over.
Welcome to Let's Get to the Marks. We're looking at an A-level biology question on cell fractionation and microscopes. Please tick a box next to the sequence that shows the correct order of magnitude of these measurements. Pause the video, have a go, then unpause and watch the explanation. Okay. Are you ready for the correct answer? The correct answer is row number 2. So put a tick there, and I'll go through now and explain how I got to that. So first of all, I've written these down into decimals, so not in their standard form. So 0.05 millimeters, that's 5 times 10 to the minus 2. Now, the next unit is in meters. If I want to also convert that to millimeters, you've got times meters by 1,000. So that gets rid of, so that's basically 0.5 times 10 to the minus 2. So that's the equivalent to 0.005. Then I've got micrometers. Okay, I want to convert those into millimeters, so I need to times by 1,000. So I've got 0.5, sorry, divide by 1,000 because we're going back up to millimeters. So you divide when you go up, and that equals 0.0005. And then I've got nanometers, and I've got 50. So I need to divide by a million, basically, by 1,000 times 1,000. So I need to divide by a million. And that puts them all into millimeters. So what I've done is converted them all into millimeters at the bottom there. So you can see they're going from largest to smallest. And so there you go. That's how we clocked onto that one. We had to go through, convert them all to the same units, and then only then can you see clearly if they're in the right order. Okay, moving on to the next question. Pause the video, have a go. You've got to label identify what is D and what is E on this electron micrograph. So what is structure D? Well, it's pointing at a stack of thylakoids. So you can either say thylakoids or a stack of thylakoids or the thylakoid membrane, because it's hard to differentiate if it's pointing to the whole thing or a group, or you can say granum or grana, singular or plural for a stack of thylakoids. Okay. They might even accept thylakoid membrane because it's hard to tell whether it's pointing to the membrane or the whole thing, or just one individual disc. Of course, here you've got intergranal lamellae, but this round lump here for E, what is that round circle? You might be tempted to say, oh, is it a loop of DNA? Because you do find loops of DNA like you get in prokaryotes, but it's not. It's a starch granule or a lipid droplet. And so that's the second answer. Let's move on. All right, 5.3. The detail shown in figure 3 would not be seen using an optical microscope. Can you explain why? Pause the video, have a go, see if you can write down 2 reasons why a light microscope or optical microscope wouldn't be able to see all those different structures like thylakoids and intergranal lamellae. And basically, light microscopes or optical microscopes are limited by the wavelength of light, which is like 200 nanometers. So anything smaller than the wavelength of light, they can't distinguish as separate objects. So we say they're limited by the wavelength of light, which of course, if you were comparing to an electron microscope, which you don't need to here, but is a lot larger or a lot bigger wavelength than a beam of electrons that an electron microscope uses. But the main points here to get the 2 marks is just that limited by the wavelength of light, so they have low resolution. And if you want a definition of resolution, you don't need it here, but it's the ability to distinguish 2 objects as separate, okay, and not see them as one lump. Great. So the main points is optical microscopes are limited by the wavelength of light, so they have a low resolution. Basically, a lot of those organelles are smaller in their width than the wavelength of a light ray. Right, name an organelle found in a chloroplast and a prokaryotic cell. So again, you might be tempted to say circular DNA. Don't do that. That's not an organelle. It's a 70S ribosome. Okay, moving on to 5.5, we have one of these biology maths questions for 2 marks with a few steps to it. Once again, if you hit the like, that'll massively help me out. Make sure you pause the video. Unpause. Check the walkthrough. Bosh. Let's go. Scientists determined the volume of a plant cell and the volume of the organelles contained. They found the volume of a plant cell is 17,500 micrometer cubed. The volume of the mitochondria in a plant cell is 262. The volume of all the mitochondria in a plant cell is 262.5 micrometers cubed. The volume of all the mitochondria and all the chloroplasts in a plant cell is 44.1% of the volume of the plant cell. Use this information to calculate the volume of all the chloroplasts in the plant cell. This should take you 2 minutes, 2 marks. Lovely. Okay, I'm going to go through the answer. So we know the volume of the plant cell is 17,500 micrometers cubed. However, they tell us here that 41 or 44.1% is the mitochondria and all the chloroplasts. So first of all, let's find out how much of the volume of the plant cell is mitochondria and chloroplasts. So we do 44.1 over 100. You can just do 0.441. And that is going to get us the answer. Smash that in the calculator. And that is going to get us 7,717.5 micrometers are all the chloroplasts and the mitochondria. Now we just want to know the chloroplasts, so we want to just take away the volume of the mitochondria. Oh my days, what am I doing here? It's like watching a 90-year-old flick through a newspaper. Okay, here we go. There we go. 7717.5, the total volume of the mitochondria and chloroplast minus the mitochondria gives us the chloroplasts on their own, which is what the question wanted us to do. Nice and easy. Hope you found that one easy. Normally the first mark's dead easy. So if you just come up with 44.1% times 17,500, you'd have got the first mark. Sometimes the second mark's really hard and needs 3 or 4 steps. This is quite an easy one. Check out my other maths videos like the one in the link here, where I go through a whole series of maths questions for A-level biology. All right, guys, hit the like, subscribe. Let's get to the marks. We're on to 5.6 here. A biologist separated cell components to investigate organelle activity. She prepared a suspension of organelles in solution that prevented damage to the organelles, described 3 properties of this solution and explain how each property prevented damage to the organelles. And it's worth 3 marks. So what 3 things did she do and how did it prevent the organelles getting damaged? Well, this is all part of your cell fractionation topic, so you should know all of this off by heart. It's part of the AS biology, but also obviously it's part of the A-level content. So number 1, use an isotonic solution. You can describe that by saying use a solution where the organelle and the solution around it are at the same water potential. And the explanation, this is going to prevent the net movement of water by osmosis, which will prevent organelles swelling up and bursting. Okay, for example, if you had a higher water potential outside the organelle and lower in the organelle, water will move in by osmosis and cause the organelle to burst. And we wouldn't want that when we're trying to study them. So that's your first mark. You only get 1 mark. I might put 2 ticks, but you only get 1 mark for each of these, so be aware of that. Property number 2, these can be in any order as well. You don't have to memorize a specific order. So property number 2, we use a buffered solution. Now, a buffer obviously minimizes changes in pH when you add hydroxide or hydrogen ions, but essentially it prevents enzymes or proteins losing their tertiary structure or their 3D shape. So enzymes don't become denatured, or other important proteins in the organelles do not become denatured, because most proteins only maintain their 3D shape in specific pHs. So property number 3, we want ice cold solution. So we want it really cold, and this is to reduce enzyme activity. And that will get you the 3rd mark. Obviously, you don't want enzymes going about their everyday business, and especially if those enzymes will digest parts of the organelle. You might have proteases and things digesting proteins within the organelle, and we don't want that to happen, so we stop the enzymes from working, essentially. All right, guys, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks. For each question or each part to the question, just make sure you pause the video. Right, 6, figure 4, an image of a fish gill is taken using an electron microscope, a scanning electron microscope. Name F and name G, these kind of plates here that are down here, and name F, the structure that's holding those plates. Okay, let's go through the answers. So F is gill filament. A common mistake is gill arch. The gill arch is below, down here at the bottom of them, holding all the filaments off of. And G, these stacks are known as lamellae. You can call them gill lamellae. Now you need both answers for 1 mark for this one, so it's a bit of a brutal. Right, 6.2, describe and explain the advantage of the countercurrent principle in gas exchange across a fish gill. So I've got a little video here you can click on if you want to know, but I'm going to go through the answer now. So the main thing about the countercurrent flow system is that water moves across the whole length of the gill in the opposite direction to the blood flow. So water's going in one way, blood's going the other way. This maximizes the efficiency of gas exchange. Now at A level we need to explore how it ensures the steepest possible concentration gradient between the oxygen in the water and the blood, the oxygen in the blood. That way, we always have oxygen diffusing from the water to the blood across the whole length of the fish gill. At no point is oxygen diffusing back from the blood into the fish gill, or is there a net diffusion of oxygen from the blood into the fish gill? So this means that oxygen is diffusing into the blood from the water across the entire length of the gill. The net movement is in that direction. And let's have a little look at some graphs that can help explain this concept. So some of you might be able to memorize the key points. For countercurrent flow, you want to say water and blood flow in opposite direction, 1 mark. This ensures the steepest or highest possible concentration gradient between oxygen in the water and the blood, and ensures that diffusion occurs across the entire length of the fish gill, always from water to the blood. They're your 3 points. However, I'm going to go through a little explanation of how we get to those points. This is the best little sketch that you should memorize, and it shows oxygen saturation on the y-axis on the left and distance across the gill on the x-axis of this little sketch graph. Now we can see that from the start of the gill, when the distance is 0, so right over on the left-hand side by the top of the y-axis, when the distance is 0, the oxygen saturation is at its highest in the water. The blue line here represents water. As the water moves across the gill, the blue line is getting less and less. Why is that? Because oxygen is diffusing from the water, from the blue line, into the blood, which is represented by this red line here on the graph. Now, obviously, the oxygen saturation, the blood is flowing in the opposite direction. So the blood starts from the other side of the x-axis. So 0 for the blood is on the other side of the x-axis. As the blood moves across the gill, the water is always at a higher concentration of oxygen than the oxygen concentration in the blood. So movement is always from the water to the blood. Now, you might say, what's special about that? Well, let's look if the blood and the water traveled in the same direction. They started at the same point on the gill and they moved together in what's called a parallel flow system. So we'd still have oxygen concentration on the left and oxygen concentration on the right. So if you think about it, the water is going to be giving up its oxygen and going down, and the blood is starting at the same point, so on the left as well. But at some point, the blood is going to gain more oxygen than the water, and the blood is going to start giving oxygen back to the water, a net movement of oxygen from the blood into the water, which means those lines will never cross. What will actually happen is, in a parallel system, they'll both level out here in the middle, where they reach 50% oxygen in the blood, 50% oxygen saturation of the water, and we get no net movement. They're in an equilibrium, but the result is that the blood does not have a high amount of oxygen saturation. So the parallel flow system is not a very efficient system for gas exchange in a fish gill. Sorry about all the knocking. That's my 3-year-old daughter banging on the door. Scientists captured a large number of 3 species of fish. The scientists measured the water depth where the fish were caught, the concentration of dissolved oxygen at all water depths, the mean gill surface area and mean body mass of each species of fish caught. The scientists calculate the ratio of their gill surface to the fish's body mass. The results are shown in Figure 5 and Table 4. Now, Table 4 shows the relative oxygen concentration at different depths of water. Or Figure 5 did, sorry. Table 4 shows the 3 different fish species, and it shows the depth at which they were caught and their calculated ratio gill surface compared to the size of them, their body mass. Okay, so we can see here that oxygen concentration seems to go down with depth, and then it pops back up again after like 750 meters. And then we can see for these 3 different fish gills, some of them have got a large surface area to body mass ratio. Let's check it out. 6.3, use the information in 5 and 4 to describe the relationship between dissolved oxygen concentration and increasing water depth, dissolved oxygen concentration and distribution of Anoplasma fimbria. Oh my days. Right. Dissolved oxygen concentration and increasing water depth. So what we can see from that is that the oxygen concentration goes down in that first graph as we increase the water depth until about 750 meters, and after that it starts to increase again until about 1300 meters, where our graph sort of finishes. Okay, and that's going to get us 2 marks. So it's a description, so you're just saying it goes down, then it comes back up. And so these are description, because the command word in the stem is description. Look at the 3 fish species. What's the link between the 3 fish species and the depth of the water where they were caught? So we can see the first one, or the A. fimbria that we're looking at, is caught in the range of 450 to 1280. It's not caught in shallow water. It's caught mainly in the deep water. So the dissolved oxygen concentration and the distribution of A. fimbria. What we find is that A. fimbria is found in a variety of different oxygen concentrations. I'd say from medium to low. So it's not found in any of the very high oxygen concentrations. So that would give you a mark if you said A. fimbria is not found in depths of water with very high oxygen concentrations. We didn't see it at like 200 meters where the oxygen concentration's high. We found it mainly in a range of low oxygen concentrations, but it was found over a range of oxygen concentrations, so we can see. Okay, 6.4, using information from figure 5 and table 4, what can you conclude about the adaptation of the gas exchange surfaces of these species of fish? So what we can look at, the conclusion we can make here is the fish that operate at the low depths seem to have a large gill surface to body mass ratio. So obviously that would make sense, right? There's less oxygen in the water. They need to maximize the rate of gas exchange. So they're going to have a large gill surface to body mass ratio. So we want to say the fish in the lower oxygen concentrations, or at depths where the oxygen concentration is low, tend to have a larger surface area to body mass ratio. Surface area of the gill to the body mass ratio. Whereas one of the fish that doesn't operate at the very low depths, it has the lowest gill surface area to body mass ratio. That's the third fish. And that's going to pick your marks up. So you're just really explaining the link between the two graphs, which we can see that if you live in a low oxygen environment, you need a large gill surface area compared to your body mass. And there we go. That's the end of that question. Pause the video, have a go at the question, and then unpause the video and watch the walkthrough. And do that for each part of the question. Use scrap paper, bust out your calculator. Let's go. This first question, 7.1, says explain how the active site of an enzyme causes a high rate of reaction. It's worth 3 marks. What you have to bear in mind as an A-level student, you need A-level terminology. Just doing your GCSE lock and key, substrate fits into the active site like a lock and key, is not going to get you the marks. They want to know how the enzyme's active site catalyzes the reaction. How does it speed up the rate of reaction? So what do we know? We know that when a substrate joins with an enzyme's active site, we call it an enzyme substrate complex. That's an A-level term that you learn, an ES complex. You learn about that in A-levels. So you can be pretty sure that that's going to be on the mark scheme. So the substrate fits into the active site like a lock and key, if you want to say that, but it forms an enzyme substrate complex. Now, this lowers the activation energy required for breaking up the substrate. However, how do we word that? The enzyme alters its active site's shape to mold to the substrate. And we call that the induced fit theory, and we learn that in A-level. Some GCSE syllabuses you will also learn that. But if you start talking about the induced fit, that the enzyme's active site actually molds itself so that it better fits the shape of the substrate, and in doing that action, that little bit of kinetic energy weakens the bonds in the substrate. So we need to talk about the bonds in the substrate being weakened. Of course, that means there's less energy to break the bonds, and the minimum required energy to break the bonds in a molecule is the activation energy. So technically, this lowers the activation energy. So the key terms here are enzyme substrate complex, waffle a little bit about the induced fit, say that this weakens the bonds in the substrate, and that lowers the activation energy, and you'll be guaranteed your 3 marks. Let's check out the next question. Okay, so we've got this little question here about the action of the enzyme catalase. I'd say pause the video and read all of this. They've got the method here. The student put 5 potato chips in a flask, added 20 centimeter cubed at 0.5 mole per decimeter hydrogen peroxide. So they've given us those variables that are controlled. We've then got the table of results here, table 5. It's got the different concentrations of hydrogen peroxide, followed by the time in seconds for 10 centimeter cubed of oxygen to be released. And we've got a blank column here for the rate of reaction of arbitrary units. Other than the one stated, give one factor the student could have controlled. So they want to know a variable they could have controlled. What have they not controlled about the potato chips? So if you check out the potato chips, what haven't they said that they kept the same? They kept the volume and the concentration of hydrogen peroxide the same, but they didn't control the mass of potato chips. So you either want to control the size or the mass of the potato chip. You might want to control the surface area of the potato chip. So there's all different things that could affect the rate of reaction. Think about the kind of factors that affect the rate of an enzyme reaction. So an enzyme is sensitive to pH. So changing the pH, you want to keep that the same with a buffer solution. And the temperature, you want to use a water bath to keep the temperature the same. Otherwise, that's going to affect your rate. The student gave the maximum rate of reaction a value of 1.0 arbitrary units. Now, this is going to be key in our calculation we're going to do. Complete table 5 by calculating the rate of reaction in arbitrary units in AU at each hydrogen peroxide concentration. Record the rates for the missing column over here. So we've got the rate of reaction in arbitrary units. Now, a real common mistake I'm going to do first, this is what I thought first. Oh, I'm just going to do the concentration over the time to get moles per decimeter per second. But of course, this is wrong. This would give us 0.78, something like moles per decimeter used up per second. But of course, we stopped this reaction when 10 centimeters of oxygen had been given off. That didn't mean that all those moles had been used up. So actually, this is an incorrect way to calculate the rate. Now, this would get you 1 mark if for each of these you said, right, well, 10 centimeters of oxygen divided by the time, 10 over 18, for example, is 0.56. So that means there's 0.56 centimeter cubed given off per second. However, again, this would not get you both marks. This would have got you 1 mark if you did that. They told us that the student gave the maximum value. The student gave the maximum rate a value of 1.0. Now, what was the one happening at the fastest rate? Well, we've got 10 centimeters divided by 6, which was the time in seconds. That is the fastest rate going, and that was at 2 mole per decimeter and 2.5, and that gave us a 1.66. So the 1.66 becomes our maximum value of 1.0. So everything else, once we calculate the rate of everything else, it has to be converted into values where 1.66 is the maximum. So basically, you need to do 10 over the time and then divide that by 1.66, because everything is in relation to the maximum value 1.66 equaling 1. So you're going to do 10 over 18 divided by 1.66, 10 over 10, which would normally be 1, but that can't be our maximum value because we've been told the 1.66 is equal to 1. So these are the values you get when you do 10 for the centimeter cubed of oxygen over the time taken to find centimeter cubed per second, and then you make them all in relation, all relative to a rate where 1.66 is actually 1.0 in AU for rate of reaction, and that's going to get you the second mark. So the first mark was quite easy. You just do 10 centimeter cubed of oxygen over the time taken to find out how many centimeter cubed per second, and then the second mark, really, you had to convert using 1.66 as 1 as the maximum value. Right, now we've got to plot a graph. So this is just testing your graph skills, okay? So, let me just, so I'm labeling the axes of the graph. That's always going to get you a mark. But I need to show you how I know that rate would be on the left and hydrogen peroxide concentration would be on the x-axis. So the independent variable is always the first column in any biology table. And that goes on the x-axis generally. And the last column in a biology table normally is the mean, or here it's the rate of reaction. That's the dependent variable, and that always goes on the y-axis. So your dependent variable is always on the y-axis. So we're going to do rate. Oh, and we have to include units, or you won't get the mark. So rate of reaction in AU, and at the bottom, hydrogen peroxide concentration in mole per dm to the -3 in moles per decimeter. So include the unit so you won't get the mark for the correct axes. Now we need to make a suitable scale. A suitable scale should take up over 50% of the x-axis and 50% of the y-axis, and these should be actually used. So we're going to use 0.5, 1.5, 2, and 2.5. That takes us over halfway up the graph paper. And then when we come up on the y-axis, we're going to just do it going up in 0.2, and that's going to use nearly the whole page. So we're definitely taking up 2/3 or more of the graph paper, so that's good. Now we just need to plot the dots on, and this gets a bit confusing when you're doing it on the PC, because I've got to scroll back up and remind myself of what the blooming numbers are. Okay, so the first plot at 0.2 mole, 0.5 mole per decimeter, I want to go up at 0.33. We calculated that. The second plot at 1 mole per decimeter, we want to go up and put that at 0.6. At 1.5, it's 0.86, so that's 3 mini squares up. Each mini square is worth 0.2 on my y-axis. And then the last two are on 1 and 1. And that's all you need to do to get the 3 marks. I'm going to attempt to put a curve of best fit through it, but yeah, that wasn't very good. Let's try again. Okay, you don't need this for the marks. It just says you need to plot the points of your process data. So they've just given you a mark for the labels with the units, correct axes, using a suitable scale, and plotting the points. And the last part to this question says, suggest a change the student could make to this procedure so that 10 centimeter cubed of oxygen would be produced in a shorter time. How can we speed up the rate of reaction? Well, look, if I went to 3, it would still be 6 seconds. So concentration's no longer the limiting factor. Yeah, we got to a point where increasing the concentration has no effect. So what else could be a limiting factor in a chemical reaction? Well, obviously temperature. Don't forget this involves enzymes though. Surface area. What else affects the rate of reaction? All sorts of things, but that's good enough. pH, because it's an enzyme controlled reaction, that's going to affect the rate if you closer to the optimum pH. So I'd say increase the temperature, but be wary not to go above 40 degrees. So if you want to say, oh, do it at 35, say that, but don't say above 40. That's going to denature proteins like catalase, the enzyme in question here. You could change the pH or surface area of the potato chips, but I'm not going with that. Okay, guys, don't forget sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You still here? It's over.
Welcome to Let's Get to the Marks. Grab some paper, grab a pen, pause the video, have a go at the question, then unpause and watch the walkthrough. This is on proteins and amino acids. All right, 8.1, describe the biochemical test to confirm the presence of protein in a solution. So what you should be looking at here for your answer is the Biuret's test. And you want to use the Biuret's reagent, which you add in a test tube to whatever sample you're testing. Now, you can alternatively say add copper sulfate and sodium hydroxide. You'll have heard of those before. Copper sulfate is a blue solution which gives Biuret's its blue color. Now, we don't need to warm it, but when you add it to the sample, if the sample contains amino acids or proteins or peptides, it's going to turn from blue to purple. I like to think of P in protein and purple for the colour it turns. You can say pink or lilac or violet, and that would get you 2 marks. 8.2. A dipeptide consists of 2 amino acids joined together by a peptide bond. Dipeptides may differ in the type of amino acids they contain. Describe 2 other ways in which all dipeptides are similar and one way in which they're different. So what I'm doing here is I'm drawing out the structure of a dipeptide. Now it tells us in the question that they're joined together by a peptide bond. So you should be able to work out which bit of this structure is the peptide bond. And you can't talk about that because it's already said it's in the stem of the question. But what 2 other ways are the 2 dipeptides or all dipeptides similar? So what things will all dipeptides have? Obviously, excluding this bit I've circled here, which is the dipeptide bond. So they've all got an NH2 at one end. Do you remember what that's called? And they've all got a COOH at the other end. Do you remember what that's called? So they all have an amine group or an NH2 group, or you can call it an amino group / amine group, AMINE. That's the NH2 group. You can say NH3 plus when it's a zwitterion. They all have a carboxylic acid group at one end, or you can say the carboxylic group. You can write the formula COOH, or you can write COO minus, like a carboxylate group. When it's a zwitterion, it loses a hydrogen. So those are all acceptable to say, and that would get you 2 marks for the 2 similarities. But there's other similarities here as well. What elements do they all contain? What do all elements do every single dipeptide contain? Well, there's 4 atoms in a protein that are consistent amongst all proteins and all amino acids, and it's nitrogen, oxygen, carbon, and hydrogen. So they have those atoms or elements. And all dipeptides will contain 2 R groups. So that's another similarity. Now, what's the difference that all dipeptides will have? The real only difference is in the R groups. You can have different R groups. Of course, you could have the same R groups. You could be a dipeptide made of 2 glycine amino acids. But for example, the R groups can be different because you could have something like cysteine that contains sulfur atoms. Excuse my spelling there, that's not how you spell cysteine. And you could have glycine, which doesn't have a sulfur atom in it. So the structure and the constituents of the R groups could be different. All right, last part to the question. A solution contained a mixture of 3 amino acids. That's key. They've put it in bold there. You must circle these in your exam because it means it's part of the answer. A scientist passed on an electric current through the solution to separate the amino acids. She placed a drop of the mixture on some filter paper, attached an electrode at one end of the paper and switched the current on. She switched off the current after 20 minutes. We can see the spots of the amino acids in new positions. Explain what the positions of the spots in figure 6 show about the amino acids. Pause the video, have a go. Okay, you can see I put a plus and a minus at the start and the end. So we know the spots have moved towards the negative side. So what does that tell you about their charge? Those amino acids must be positive because they've moved off the line. If they had no charge, they'd stay on the line. If they were negative, they'd move towards the positive end. So they've been repelled by the positive electrode and attracted to the negative electrode. So that's going to get you a mark for explaining why they've moved that way. What can we tell by the fact that they haven't moved the same distance? There's 2 amino acids there that are separated. Well, we can tell that 2 of the amino acids must have different masses or different charges as they have moved at different rates through the filter papers, different speeds through the filter paper. So after 20 minutes, they haven't been going at the same speed. One has gone further than the other, so it must be lighter, or one has traveled slower, so it must be heavier. Now, the third mark is the hardest mark. Remember, we circled 3 different amino acids. How come there's only 2 spots? If there's 3 amino acids, why is there only 2 black spots? And that is because it tells us that 2 of the amino acids must have the same mass and the same charge because they have moved the exact same distance. So we can only see 2 spots. It means one of these spots, there are 2 amino acids in it. So obviously, that mark is quite hard to work out, but they put the little clue in bold at the top there, so you always got to watch out for that. So what I'm going to do here, I'm going to draw the black spot on the line, and what this shows us is that this spot moved, I'm just trying to select it, this spot moved the same distance as one of the other spots, so it appears like there's only 2 amino acids, but we were told there were 3, which means one of these spots contains 2 different amino acids. That must have the same weight and mass. Okay guys, thanks for watching. Let's get to the marks.
You're still here? It's over.